Wendy Hartsock, science and peptide enthusiast. In this episode of Exploration Science, Professors César de la Fuente from the University of Pennsylvania and Fabien Plisson from CINVESTAV discuss data science and peptide discovery. They also dive into their exploration of antimicrobial peptides. Well, thank you both for agreeing to do Exploration Science. I do appreciate that. Hopefully you'll have fun.
It's my pleasure, to be here, sharing some time with both of you.
Fabien, could you talk a little bit about what your current research interests are, and like what your lab is actively working on, and maybe even what you're sort of hoping to get into?
All right. So basically, my lab, so I'm basically in the center of Mexico, so 5 hours north of Mexico City. So my lab mostly focus on three different areas. So one is the discovery of bioactive peptide using like venomics and proteotranscriptomics on one side. For the students that are interested mostly in hard-end, like doing lab work. And then, but I'm also mostly interested in, and that's a 10 years old journey in implementation of artificial intelligence and machine learning algorithm to the development of discovery and design of bioactive peptides. One is the antimicrobial peptide, and that's where we join with César. So it's mostly, that's because that's one of the databases that is the mostly populated. So it's one of the easiest, let's say, to start to be able to build up both predictive and generative models. And and then the other one is basically the bottleneck at the moment in my lab. It's the expressions of those peptides in system organism or other synthesis. So they mostly focus both on venom secretions on one side, and then the others plant and antimicrobial peptide sources in general. Yeah.
Perfect. Great. And then César, could you also do the same, talk a little bit about what your current research projects are?
Yeah, definitely. So in my lab, we focus mostly around the use of computational tools to allow us to accelerate antibiotic design and antibiotic discovery. So we use, an entire toolkit of tools. This includes also predictive models, generative models, and with the dream of eventually being able to discover antibiotics that will help us, that we'll be able to transition them to actually helping people. That's kind of the dream that we have. And in order to do that, we have in the lab, we have both computational expertise and also experimental expertise to be able to validate experimentally the computational designs and the computational assumptions, right? Of course, we're not at the stage in this field, this very much emerging field, where we can just trust the computer. And so we absolutely, typically, whatever designs the computer creates, we synthesize those and we validate them to see whether the computer was correct or not. And then we have a number of mouse models that are of preclinical relevance that where we also assess the anti-infective efficacy of anything that we create on the computer. So we can actually test whether whether we see what we see on the screen is actually useful at killing bacterial pathogens, mostly in vitro, and then in mouse models that are relevant. So that's kind of what we're most excited about these days.
Excellent. So a couple of questions around this. I know this is something that gets brought up a lot, of like how many peptides do you need to test from your predicted models where you feel confident that your models are interrogating, I guess, the space enough?
So we try to generate the peptides to build our own data sets in-house. We think that's important just to have a more reliable source of data, as opposed to, with databases, they're obviously very useful, right? Because you have all this information in databases, but oftentimes those databases, they are generated by different labs using different media and different conditions, and so that makes it extra hard on the computational side to be able to extract anything that is like really useful. I'm not saying it's impossible, of course, but so we try to build our own data sets in the lab, where by synthesizing and experimentally testing everything, using standardized conditions. And then that's what we use as our training sets. Sometimes we do have to supplement our training sets with database sources, but anyway, that's kind of how we're thinking about it.
Okay, yeah, I guess I asked the question weird. What I meant was, so say you've computationally made, I don't know, 1 million peptides.
Mm-hmm.
How many do you test out of that set?
Oh, yeah. Mm-hmm.
Try to do the minimum, I guess. The idea is to show the strength of the computational approach. So you want to be able to get, let's say, to do the whole idea of computational or computer-aided algorithm is to minimize the number of steps and to the consumption of resources. So the idea will be to do the least number of peptides to prove your point.
Right.
And maybe 10, 20 maximum to prove your point with the highest probability of being non-hemolytic, anti-cancer, things like that. So that I think that's the whole idea is to minimize the number of steps and limit the resources associated with it by using human knowledge, let's say papers and databases that have been documented so far.
So I guess, yeah, as the field's growing though, where do you, because you're still trying to figure out how to get there. That's the goal, right? Is to have minimal. So César, when you're looking at this problem, how do you approach it?
Yeah, I agree with what Fabien said. We validate on, we're also limited, but obviously by the cost and the time that it takes to this, synthesize the peptides and do the screenings. But we do, dozens usually. In some of our more recent work, maybe we do like 100, but it does get expensive. And so yeah, I think in the end, I think that the goal is where it's a future where we have peptides that are reliably predictive in terms of the function that we expect, and that we don't have to synthesize a lot of them because the algorithms that we're using are actually very reliable, and doing so with minimal human intervention. I think that's kind of the goal. Obviously, we're not there yet, and like I mentioned earlier, we very much need to absolutely validate everything nowadays, but I think that's where we should aim to head towards.
Mm-hmm. Where, for both of you, where are you finding the most interesting starting point to look at antimicrobial peptides? So fruits, venoms, other areas. Fabien, go ahead.
Well, usually those antimicrobial peptides are documented in databases, and you can have like a focus on certain sources like plants, things like that, if you are interested in certain family of peptides. But depending on the structure as well, you're interested to focus. So some alpha helical structure will be found in insects, whether the disulfide-rich peptide will be found in plants. So that could be a bias that you can choose to take. But usually when we build model, we don't look at necessarily the taxonomy per se. We look at the distribution of amino acids, and you could, depending on, I think I mentioned it in last week talk, that you could eventually remove certain sets like peptide rich in prolines or cysteines because they're complicated to make. Therefore, you bias your algorithm towards certain structure. But and also in some way the taxonomy or the taxons you will be looking at as well. So yeah, I don't. Yeah, in that case, when you build model, I don't necessarily pay attention to the sources they come from. Mostly the structure.
Okay. Is that how you approach it, César?
Yeah, I think structure is obviously super important, but I think sources, we're not limited by any source in particular, and now with the availability of genomes and proteomes from numerous organisms, both existing and extinct organisms, I think there's a huge potential for exploring just biology, right, as an information source of code of nucleotide code for genomes or amino acid codes for proteomes, to try to find sequences that encode for peptidic structures or peptidic sequences that are that could be antimicrobial or could be anti-cancer or numerous other potential functions, right? And so I would say, you can take the structural approach where you filter using structural components, and you can, but in terms of, in terms of looking at organisms, I think we're not limited by anything, as long as we have the genomic or proteomic information that we need.
Sure. I guess I'm kind of curious to know if there are certain, if you look at different species of, like, say, wasps, you might say, oh, well, these wasps tend to be resilient against a certain type of infection, or some other, like certain things in the animal kingdom that you see that they're more resilient to certain diseases. Is that anything that you've looked at computationally to see if those folds are different, or has anyone looked in that area, or is that even, I don't know, an unintelligible question?
Well, at the moment, I'm looking at the venom of ants, so related to wasp. Yeah. And then we actually testing them towards Candida, because many ants also have defenses against fungus. And therefore, there is some sort of relationship, like the one you mentioned. Yeah. So we're interested to see if we can find antimicrobial or antifungal peptide from those sources and test them against Candida or Candida albicans in that particular case. So, yep, that's really your question.
Nice. That's great.
Yeah, that's interesting to think about that, right? Where, I mean, and how obviously a lot of these molecules, they play a role in host immunity, and so to try to investigate that further, I feel like we still don't understand a lot of it. Like, if you look at venoms, a single venom can have 1,000 different peptide sequences in it, and we don't understand. We haven't explored even a small portion of that, and so I think there's still a lot to be learned, and that's obviously part of the excitement of this field, of peptide research and peptide science, is that there's so much to be explored, and a lot of this, and that's only looking at nature, right? But then, with computers, you can start exploring previously unexplored regions of sequence space that have not been sampled throughout evolution, and you can try to find interesting molecules there as well. So I mean, I think it's a very exciting time for the field.
Mm-hmm. What do you currently think is the biggest barrier to developing antibiotics for mass use?
Well, that's a
I know
multifactorial answer there, because I mean, there's no market, right? There's not enough incentives for an antibiotic drug to be profitable. In fact, we've seen companies that have successfully taken an antibiotic all the way to FDA approval, and those companies have eventually folded. And so the market is not receptive of antibiotic drugs for many reasons, and that involves policy. It involves incentive, changing the incentives, cultural shifts, and those are things that people are working on, and we are making progress on. And then there's also an innovation gap, where for decades we have been unable to discover entirely new classes of antibiotics, and that's, I think, where computation and AI come in, where I think such tools can help us accelerate the discovery of really truly new classes of antibiotics. And I think that's what I'm most excited about. And but again, I don't know if Fabien has any thoughts on this.
No, yeah, I completely agree with what you just said. I mean, it's cultural shift. There is a lot to take in. I mean, right now with the pandemic, I don't know if it's done or not, but people seem to reconsider the possibility of treating and to reconsider, I mean, to use of antibiotics and abuse of antibiotics, and see if that take any against viruses and other pathogens, and see if that will be changing our behavior, let's say, and practice with those. But I mean, you summarized very well. It's a complicated area. I mean, there is also a consideration at the moment of pathogens in crops. There's a rising among the monoculture. There's a rising number of fungi and bacteria and virus that plague pretty much our common crops. And here as well, there is potential opportunities to use antimicrobial peptides for treating those issues.
Yeah, that's a really good point. I think agriculture is often overlooked as a large area for peptide science. I know David has some work with cyclotides to help with pest control, but yeah. So I guess something that I find very interesting, Cesar, you had brought up the immune response, and I know that you've worked with, I don't know what you would call them, I call them like grafted peptides, where you have an immunomodulator and an antimicrobial agent. Something else that I find very interesting, I think both of you have done work in this in the microbiome and signaling. Are you working to look at, I guess, other mechanisms of action? So instead of like targeting a bacteria specifically, instead targeting the commensals in and around an infection to stop like the bad bacteria, the pathogenic bacteria? Or have you guys seen work on that? Is that a...
Yeah, I mean, I think microbiome engineering is a super interesting field. Of course, the microbiome, in particular, the gut microbiome. So these are like the indigenous microbes that live in our guts and have co-evolved with us. They have been associated for the last 2 decades or so with many, many diseases, from cancer to obesity to even neurodegenerative diseases. And the issue is that those are all correlations, but we don't have technologies or the technological tools to allow us to interrogate microbiome systems to be able to identify whether those correlations are actually causative, whether bacteria A and B are actually causing disease X, right? And so I think peptides and other molecules can be great potential tools to allow us to elucidate causality between microbiome members and different human and animal diseases and plant diseases.
Sure. Okay. Well, so then maybe going back to your peptides, the immunomodulators, can you talk a little bit about those, just for people who maybe don't know?
Yeah, absolutely. So I think, the way you traditionally treat infections is with classical antibiotics, that what they do is they target the pathogen, you target the microbe, you destroy it, and then that's how you clear the infection, right? But there's another sort of paradigm of thinking about infection clearance or treatment, which is by, instead of targeting directly the microbe, why don't we try to target the host? So ourselves, or in our case, mice that we use as models for ourselves. And by targeting the host's own immune system, you can boost immunity. So then the actual animal can clear the infection by having sort of like a super immune system. And we've done a bit of work where we've combined those two activities into dual synthetic compounds or molecules that can do both. They can directly target pathogens and they can boost the host's own immune system. And so you have a two-pronged approach to treating infections. And again, immunotherapy is obviously not a new concept. It's been used for cancer treatment, and it's been used in virology as well. But for the treatment of bacterial infections, it actually is a very new sort of concept. And so I think we're excited about that, and we're continuing to pursue those avenues in the lab.
I have another super basic question. Fabien, the venom peptides that are known from, all different species, what is their actual use in those animals? So these ant venoms, why are the ants actually creating antimicrobial peptides in their venoms?
Well, I guess it's to protect themselves in the first place. No, so there is, like you said, the venom is usually complex. There is toxins that are used for predation on one side, or protections, and the others is to defend themselves against potential predators. So it's one part of their own protections against potential predators and infections to protect themselves. Yeah.
Okay. The reason I thought of that, sorry, I was just thinking of like those mixtures, obviously there's lots of things going on, lots of cooperativity between the different molecules and venoms to help like kill whatever they're trying to kill or paralyze whatever. Yeah, it was just because it's the antimicrobial peptides would have been sort of concentrated in the venom sac, right, compared to the rest of the animal. So Cesar, I don't know, Cesar, did you have something to add to that?
Yeah, I think there's a lot to uncover there. I think we're finding in our own work, we've performed the first proteome-wide exploration of the human body as a source of antibiotics, and we're seeing all sorts like thousands of small fragments that get released from parent proteins, and that might be the same in other organisms such as ants or scorpions or snakes, where we're actually having all throughout our body these little fragments getting released, and they cooperate. They when you combine them in cocktails, they sort of cooperate with each other to target pathogens. And the way we're thinking about this now is that it's perhaps perhaps part of immunological response, right? And some of these fragments, they come from proteins that are not part of the innate immune system, which is fascinating, I think. But they come from the nervous system, the digestive system, cardiovascular systems, and the way I imagine this in our bodies is, again, this is just a hypothesis, but that we have all these proteins that have their day job, right, in the nervous system, or making sure homeostasis occurs as it should. And then under certain conditions, we have chewing enzymes that chew up the protein. These are called proteases, and they chew up the protein, releasing peptidic fragments that we call encrypted peptide antibiotics. And we have like thousands of these in our bodies, and when a particular invader takes over our bodies, let's say a virus or a bacterium, perhaps these things are getting released all together at once or over a time scale, and they're helping sort of counter that particular pathogen that can be dangerous to our health. And I don't know, that's kind of how we're imagining that this might be happening. But again, we're still sort of investigating this and trying to trying to see if that's the case,
Sure. So are you looking at like particular infections, like actual, like a wound where you would then decipher what peptides are co-localizing there? Or how are you interrogating that?
Yeah, what we've done is we've identified different sequences that are encoded in proteins from different areas of the body. Let's say the bloodstream or skin or the brain, etc. And now we're trying to characterize those as much as possible within the context of where they are located. And so if you think about blood, you can think about a sepsis sort of scenario where you have a systemic infection and the bloodstream is infected. Do you get better clearance of those infections when you treat with those particular peptides or not? And that's kind of how we're thinking about it. But we're still very much in exploration phase of all the data that we've been able to generate.
All right. See, yeah, it's the question, I think, join a bit the question you had earlier, whether we can observe and apply peptide to microbiome, because most of the knowledge right now has been tested against a handful of strains, bacteria strains. And so if we build our machine learning models, they tend to be built on certain strains. And we eventually assume that the activity could be similar or different on the other strains. So it's hard to really figure out. We can build a robust algorithm for E. coli, Staphylococcus aureus, or Pseudomonas, and then the disease related to it. But then when it comes to figure out what sequence are the right one to only target Staphylococcus and not targeting E. coli, it's another story, because it's not always easy, because we just learn how to build those models for one particular predictions, and so crossing the data, it's a current challenge at the moment. And I think what César was talking about when he was talking about peptide with dual activity, same thing. You on one side you predict immunomodulation, and on the other side, and you want to maintain antibacterial activity, and you want to figure out if you can combine more than one algorithm and build a sequence that combine both predictions, or both reliable predictions for it. So it depends really on what we know at the moment and what we can do with what we know for the potential application.
Right. Yeah, I think this is something that I brought up with those computational lectures that we did well, you guys gave them, I just hosted it, with Ava about the neoantigen world was that so you have this area where you're predicting peptides as vaccines against solid tumors, and you're using these neoantigen sequences to do it. And so you do these predictions, you make these peptides, they seem to work at least in vitro, and then you put them back in and they're not actually active against the tumors. And to me, it's not necessarily that the sequences are off. We don't know, right? It could be that the way that those sequences were presented back to the body was off. So this is why I think a lot of companies were moving towards actually engineering the T cells and putting those back in, but that's of course incredibly expensive. And so I think when I hear you talk about these areas, I think there's so much to not just the machine learning part and the peptide prediction and is it active, but is it active against a certain bacteria? Is it active against that bacteria when it's in a certain organ? Is it active against that bacteria when there's a co-infection? I mean, there's just so many layers of, I guess, complications. And in the empirical world, you just test it, right? You go into the wet lab and you're like, you inject a mouse with, some extract and you say, oh yeah, it has this behavior. But in the computational world, how do you start, I guess, targeting those different factors?
I mean, you rely on data too. So, in the end, you can't get away from the data generation part, I don't think, or the experimental part. But, computationally, what you can do, you can apply little tools like transfer learning to try to transfer knowledge from one domain to another that, might have applications in what we're talking about. For example, if you want to transfer data from, I don't know, different concepts like antimicrobial activity, and then you want to look at toxicity. You might be able to do that in that way. There's also sort of multitask learning, where you can look at different tasks at once simultaneously, and those are some things that come to mind. But in the end, you can't get away from the data. Again, the machine is just learning what we feed it, and what we feed it is a training set that is composed of the data generated either by others, data integrated in databases, or data generated in-house. I mean, and so those data need to be as reliable as possible and as predictive, as robust as possible. But Fabien, maybe you have other thoughts here.
Oh, well, yes. I mean, it's like you said, it's about transferring the knowledge from one area to another. I mean, right now, so we start building maybe 5-6 years ago, the very first model on single activity. Now we start playing to combine those, but there is a risk that the domain, the data we learn from one data predictive model and the other do not overlap, and that's becoming very complicated to be able to combine those models together for predictions.
Yeah,
that's a risk. Then we need to be aware, etc. And then you can combine. I mean, the whole strength of machine learning is that you can explore a given space of peptide sequences and apply as many models as you want. The same way that when you do medicinal chemistry per se, you focus on inhibition and then binding affinity and then solubility, and then I mean it's you get so many markers or metrics that you need to consider in parallel to get the right fit or the optimal compounds. So the same optic there, but using data set that are existing and our own biases, because historically we focused on certain type of structures. We focus on certain time. You were talking about David Craig earlier with cyclotides. So obviously there are databases that filled with a certain type of activity. I mean, if you want to look at immunomodulation, catalycidins are probably one of the most studied peptides in this area for the so we need to take these in considerations to build our models and to be aware of it.
And I think eventually. Oh, sorry. I was just going to add, like, to create a drug is a very complicated thing, and you in addition to just optimizing for antimicrobial activity or whatever function you're interested in, you need to take into account so many other layers, including we discussed stability, to make sure you're particular peptide or molecule is stable enough so that it persists in the body to exert its function, it needs to be non-toxic against our own cells, human cells. It needs to be, it needs to have biodistribution profiles that are appropriate. So PK/PD, thinking about that. And so it really is like many parameters that are very different from each other, and yet you need to build one small molecule really that contains all those desirable things. And again, it's a very difficult thing to do, and obviously that's why it takes so long to develop drugs, right? On average, over 10 years. And that's why it's so expensive, on average, over $2 billion to develop a drug. And I mean, of course, that includes clinical trial costs, but even to discover a drug is a very, very costly process. And so yeah, it really takes a village to think about this. It takes people from different areas and domains working together. It takes interdisciplinary teams working together. And I think that's part of the beauty of it. When you're trying to tackle something so big, so difficult, you really need to cooperate and collaborate with others.
Mm-hmm. Do you think that right now, so I've also heard like Ava List from Caliber was talking about her success using a very small data set with antimicrobial peptides. And I guess some of the things that she said made me wonder, like, in general, is this a field that is more amenable to AI and ML compared to some other areas? Like, is there something unique about antimicrobial peptides in particular that sort of make them more, I guess, easily studied through computation?
Well, I guess it's more the rise of natural language processing models and language models that are both applied to peptide design and protein engineering that make it not only antimicrobial peptide. You can think of any peptide or any proteins family with a given focus. I mean, we could easily translate this kind of technology to the design of new venom toxins or derived synthetic toxins, or it's already done, but immunoinformatics is the engineering of epitopes. You got. I mean, there is a lot of translation that can be done. Yeah, I mean, and AI is also applied to small molecule now. I mean, you get graph neural network that done to small molecule as well, and natural product. So yeah, it's a very interesting, let's say it's a very interesting, help a lot to digest the whole, let's say the whole history of drug discovery, and then to make it more digestible and understandable. And then today we can actually process data very quickly and be able to see those trends or to see those things that we couldn't see before by just opening an Excel sheets. Now you are able to digest much faster this sort of information, and antimicrobial peptide is one particular niche area when we see potential to develop antibiotics and potential for health, let's say.
Yeah, the way I think about peptides is, we have the obviously the dogma of molecular biology, right? We have DNA, we have mRNA, and we have proteins and peptides. And if you think about it, DNA is programmable, mRNA is now programmable, but peptides and proteins are not yet programmable. So I think it's the remaining sort of, out of all of life's molecules, within the dogma of biology, they're the last subset of molecules that are not yet programmable. So I think that makes them extremely exciting. And the other thing is that, of course, because they're part of that, they're encodable in mRNA, they're encodable in DNA, and so you can find them in genomes, you can find them in proteomes, you can find them in mRNA data, and that opens up many avenues for discovering novel peptides. I'm not talking only about antimicrobial peptides. I'm talking about any sort of, like what Fabien was saying, any kind of peptide that might be bioactive, that might be interesting from a therapeutic or other perspectives, and that's why I think peptides are exciting. And with the same goes with proteins, in my opinion. I mean, now we have AlphaFold that can predict the structure of proteins. it doesn't work very well for peptides, and which makes a lot of sense, of course. AlphaFold was not built with a training set composed of peptide sequences, but I'm sure it won't take long for DeepMind or others to figure out how to predict peptide structure. But anyway, there's just a lot going on, and again, it's the next frontier. mRNA, we can make vaccines now. We can make therapeutics out of mRNA. Obviously, with the help of lipid nanoparticles to prevent degradation, we can do that with DNA. But I think a number of things need to happen in the peptide field. One of them is decreasing the cost of peptide synthesis. That's what we saw with DNA synthesis, right? And sequencing is there was a huge decrease, and that's what enabled a whole field to be opened up, and it enabled DNA to be programmable. So we still are not quite there in the peptide field, and then try to understand more from basic principles the underlying chemistry and physics of amino acids, and not only in isolation, but when you put them together within a peptide sequence, even a simple like linear chain, how do to be able to program things, you need to be able to understand them perfectly, and if you mutate a particular amino acid, you remove it, or you add a particular amino acid, to be able to predict what the function is going to be. We're not there yet, obviously, and again, biology is complicated. It's chaotic. It's not a precise science like mathematics or physics, and so that's why we need to take into account many things, including physics, chemistry, mathematics as well, modeling, to be able to try to understand a biological system such as a peptide molecule. But I mean, all these things are super exciting because they're not known.
Yeah, absolutely. What are you hoping to see as maybe the next big breakthrough in computational design of any peptides? Very open question.
Yes. Yeah, you want to go first?
I can try. Let's see. I think AI can help accelerate building peptide molecules that can be potential drugs. But again, we're still learning how to sort of walk. Let me just put it that way in this field. It's a very young field. I mean, if you look at the number of papers published, it didn't even exist really like 5 years, 6 years ago. And in terms of, critical mass of people working in this field. But I think, maybe developing a technology that drastically lowers the cost of peptide synthesis would be huge for the field. Because again, for proteins, right, you need to recombinantly make them, and we're also not very good at doing that. It's very sort of, I mean, Fabien, you were referring to that earlier, right? But it's very case by case scenario, and there are a lot of unknowns. I'm not sure why this protein is not getting secreted from the cell. We're talking about recombinant production in microbial cells, so E. coli or Pichia pastoris. But in peptides, we can synthesize them using a machine, right? We're using solid phase chemical synthesis. So I think one huge breakthrough would be to lower the cost of that and the accessibility of that, so that then, like now, you can order a primer, a DNA plasmid or anything like that, and it's incredibly cheap. Anybody can get it, highly accessible. I think that's what we need to create in the peptide field. And then I think AI can accelerate, again, building molecules that look like drugs that potentially can enter clinical trials and then build momentum in the field, right? Once you start getting enough preclinical candidates into phase 1, then the field starts growing, and hopefully we see that.
Mm-hmm. Yeah, to bounce back to see what César was talking about, AI technology. I mean, when you think about it, every single protein engineering or peptide design is built upon the relationship between sequence, structure, and function. And so AI, AlphaFold is just one of those predictions between sequence and structure. But then you also have technology that are currently being built to directly predict function from the primary sequence. I mean, BLAST is one basic one, but there is more complicated architecture that are being built right now. There is structure and function. I mean, it's not that good at the moment, again, structure input, and because two proteins that could have the same structure could have different functions. How do you predict that? How do you know that you obviously can test it. I mean, right now we have limited information, so you will have to test it, but you could eventually using AI technology. So that will be a breakthrough to advance our understanding and to potentially try to limit our exploration when we get all these next generation sequencing with a lot of transcriptomic and very few sequences. Then this gap basically could be actually reduced by AI technology that not only focus on structure but also on functions. That's another paradigm to be changed. And like César said, I mean, any lab technology can improve and reduce the cost of rapid productions of peptides and design. I mean, right now we rely either on solid phase peptide synthesis or protein expressions, and yes, it's a nightmare.
Yeah, I couldn't agree more. And relating structure to function is still a huge it's a huge challenge that hasn't been solved. And I would also add that dynamics of peptides and proteins, if again, going back to AlphaFold, I mean, that it's just a static prediction of a static protein. But the next step is to also look at the dynamics of proteins and peptides and try to predict that, right? Either through molecular dynamic simulations or other types of simulations. And because dynamics are important, right? Like if you think about a peptide, right? If you take a snapshot of a photo of a peptide at a given moment, the peptide is jiggling in space nonstop. You take a photo, you get this conformation, but that gives you 15% of what the peptide looks like in a particular moment. And so gaining insights into dynamics, I think it's important both for peptides and also for proteins, and probably that's going to be the next step of protein structure prediction and hopefully peptide structure prediction as well.
I will say, talking about like the fact that it's a new field, so there's all these areas that we're getting to know more and more. I am seeing more and more large companies advertising jobs specific to AI, computation, molecular dynamics, and so hopefully, as the community sort of like reinforces the number of folks working in this space, we'll have some breakthroughs as well. And then also, obviously, companies are interested in applying this to like further downstream, so to clinical trials. Right? So hopefully, some of those aspects of it will help accelerate the utility of these techniques. So I want to take a step back. Because we started out with sort of like what research was going on in your laboratories, but I'm always curious to know like how people got to where they are. So maybe I'll start with you, Fabien. What was your PhD in?
Marine natural product. I was diving. I was extracting sponges.
Oh,
That brought me to Australia actually. I was actually working under the supervision of Rob Capon in Australia, and he shared the same institute as Glenn King and David Craig. So for the little story, so I knew those guys because I spent both my PhD and postdocs in Australia, listening to the talks of those guys, talking about venoms and cyclotides. And so if you want to connect with the AI part, is that 2011, 2012, I was a bit frustrated to explore natural product diversity. And then so I get into doing basic statistics like PCA to represent diversity. I mean, in that time, most people were focusing on molecular diversity using unsupervised technique. And then few were doing supervised learning, like machine learning model that we apply today. There was very few of them. They came a bit later. It's also the time that the MOOCs, massive online courses, started to happen. I mean, Coursera, EDX, things like that. So because my background was mostly organic chemistry, I had to train myself using all these online courses in machine learning. I mean, that was the data analysis from Johns Hopkins University at the time. And then so I basically start doing this so I could build up my very first predictive models, applying those binary classifiers to marine natural products. So my PhD was mostly focused on finding new kinase inhibitor to treat Alzheimer. And so one of my peers basically asked me if I could pass the blood-brain barrier. And I was like stuck, but my boss didn't want to send any money to do lab testing. So I said, okay, let's do in silico stuff. And so I learned from scratch, basically doing machine learning. I very enjoyed the logic behind it and the rationale of building models. So, and then I moved to peptide because I felt that if I wanted to explore more the chemical diversity, peptide were easier to synthesize than natural product. So, I mean, small molecule natural product. So I went and I was like, yeah, I can explore that much. So I went and worked with David Fairley and David Craig, and in collaboration with Pfizer, working on GLP-1 analogs, doing full lab synthesis and a bit of computational work. And it's afterwards that I went and merged the two fields when I create my lab, basically.
Awesome. All right. And César, how about you? What was your PhD in?
Yeah, so I did my undergrad in biotechnology, bioengineering, and then PhD microbiology and immunology. I did it at University of British Columbia in beautiful Vancouver, Canada, and with Bob Hancock. And that's where I started basically exploring how bacteria become harmful to humans, and trying to sort of interrogate the different mechanisms that they utilize to make them successful, right? To colonize our bodies and to sort of thrive in our bodies, in our environment. And towards the end of my PhD, I started playing around with actually different peptide sequences to see if they could be antimicrobial. And I was fascinated by the fact that some of them were able to completely clear the bacterial cultures, so completely kill bacteria, right? But at the time, I was frustrated by the fact that in order to engineer peptides, you have to manipulate them one by one, and it was a lot of trial and error. You have to resynthesize the molecule just to generate one mutation, and so on. And so I wanted to, for my next step, I wanted to try to generate tools or learn tools that help me program peptides in a more systematic way. And so then I went to MIT for my postdoc, where I worked on synthetic biology and computational biology. And that's where I started delving a lot more into how to program peptides systematically using machines. And then after that, I was lucky to be recruited where I am now at University of Pennsylvania, UPenn. And basically in my lab, we've sort of converged all those different concepts into what we do in the day-to-day. So we have computational work, we have experimental work in vitro, and then we have experimental work in vivo using preclinical mouse models. But that's a little bit of the background. I've always just been following sort of curiosity and trying to learn what I thought I needed to learn to solve the questions that I wanted to solve at each stage. And when it was all trial and error, it's also boring. I got bored by it, and I thought, okay, it's frustrating, right? It's like, okay, this is not engineer. Like, you cannot engineer this if you're just like shooting in the dark. Like, okay, let's make this mutation and this other mutation. Well, what is the rationale behind that? So sort of using more like first principles thinking around that, and I think computation helps bring some of that rationale to a lot of what we do now.
Right. Yeah. Well, I have a couple more questions, but before I do you guys have any questions for each other? Or maybe you want to talk about something that I haven't asked a question about?
Well, I mean, I think we've talked already, but I'd love to collaborate. So, we're interested in venoms too. And if you guys need any help with, I don't know, in vitro, in vivo testing or anything like that, I'd love to explore possibilities of potential alignment in some of the work that you're interested in.
Sure. Happy to help. We built some models, so we need to do some protein expression, and so with the pandemic, that has been a bit slowed, right time. But now we can explore that. There's a lot of questions open, like whether we can use, for example, machine learning to understand better or to predict binding interaction of those toxins with ion channels. Like that would be a great thing. I mean, then we can support that afterwards and to further understand and to figure about the selectivity towards those ion channel subtypes. That would be even and design possibly some of the toxin towards one or the other. So those are fun questions to answer with the current data that we have. I mean,
yeah. Well, happy to explore offline whenever you want.
Yeah.
Yay, collaboration.
Yeah, no, I mean, that's great.
No, I was just gonna say, Wendy, bringing people together here, and thank you for having us in this really fun, sort of interactive and informal conversation. It's really enjoyable.
I love it. I can't get enough of it. It's super enjoyable for me, especially in fields like this where it's like, I studied AMPs as a postdoc, but not computation. I was interested when I was an undergrad. I picked up a C++ manual and very quickly put it down. I was like, I'm going to teach myself C++. And I was like, no, I'm not. But something that I am curious about the venom world, Fabien, when you're looking at different sources of venom, where do you see the most overlap and where do you see the most divergence? And again, I know these are kind of like very broad questions, but like if you look at an ant versus a wasp, you would expect they'd be somewhat similar. But are there cases where you see like ant versus platypus?
Well, you do see convergent evolution in some species. I mean, recently they identified one of the very first plants in Gympie in Australia with needles, and the toxin found in there had similar structure to the one from jellyfish. I mean, it's quite funny. I mean, to figure there is some convergent evolution at the molecular level. I mean, here you can. There's a couple of work from Ivan Udheim and then Glenn King towards looking at toxins as molecular probes for evolutions and looking where do they match between different species. So it's a very interesting area to figure it.
Right. Well, and I'm sorry, now I'm going to skip to another question, but César, one of your projects was, looking at you were doing like evolution of these antimicrobial agents, and what you found was that if you went too far, you kind of lost some diversity. Or can you talk a little bit about that?
Yeah, sure. That was a collaborative project where we developed this evolutionary programming algorithm to allow us to evolve peptidic templates from nature. And the way we did it is we trained the computer to execute essentially Darwin's algorithm of evolution. So the different steps of evolution, of course, biology 101, right? Like mutation, selection, recombination, and then you can loop that in a feedback loop, and essentially you get evolution in silico in real time. And the computer was able to actually design peptides, evolve peptides to make them, to optimize them for antibiotic properties that we then were able to validate in vitro and in vivo. And that was actually, I think, the first description of a peptide antibiotic that was effective, that was designed by a computer that was effective in a preclinical mouse model, and which was very exciting to us. But the way the algorithm, so going back to your question, so if you let the algorithm run for too long to reach an optimal solution, what it eventually did, it started spitting out sequences that looked very similar to what we see in biology. However, we obviously wanted to generate diversity or innovate at the peptidic level, at the sequence level. And so what we did is we stopped the algorithm before it reached that optimal solution. So it was really a suboptimal solution. And that's where we got diversity, and in terms of sequence, in terms of amino acid ratio composition, and so on. And those are a lot of the peptides that we ended up synthesizing and validating in vitro and in vivo were generated through those means. Philosophically, I'm not sure what that means, but basically it was a tweak on the algorithm in order to be able to really come up with new sequences. And since then we've now moved on from evolutionary programming, and we're doing a lot of deep learning for a lot of our work. And we have a number of things in the pipeline, which I hope we get published soon and people can read them.
Awesome. Great. Well, yeah, we have like 1 minute, so maybe I'll just, I think I could keep talking to you both for like hours, but maybe we can have a part 2 at some point. So I'll just say thank you so much for your time. And yeah, I appreciate it.
Thank you both. It's been really a privilege spending this hour with both of you. And yeah, I hope there's more in the future.
Yes. Thanks for tuning in to this episode of Exploration Science. As always, we welcome your feedback and suggestions for topics that you'd like to see covered. You can leave those suggestions in the comments below or tag us on LinkedIn. You can also find this podcast available as a YouTube series by searching Exploration Science. Thanks again for tuning in.